So the last video, sorry, a cutoff, I'm going to pick up at that same point. Looking at the other components of the blood, the first one that you have is the white blood cells or the leukocytes. Leuko indicates bones, um, and cyte indicates cell. It gives you a list of the 5 most common types of white blood cells that we have. We are not going to go through those right now. That gets picked up in the lymphatic chapter. The blood platelets, those are referred to as your thrombocytes. And thrombocytes are actually going to come from a cell that is known as a megakaryocyte. And honestly, that entire name is a bunch of prefixes and suffixes all sandwiched together. Mega indicates large, karyo indicates nucleus, and cyte cell. So it's telling you that you have a very large nucleated cell. Now, normally when a cell dies, it is going to get sloughed off somehow. Your skin cells flake off, other cells leave through your waste. But with the megakaryocytes, they do something very different. Instead of getting sloughed off or instead of getting digested, they are actually going to break. And if you think about what would happen if you took a glass plate and dropped it on the floor, it would shatter into a bunch of little pieces, and you would have all these sharp, fragmented edges all over the place. And that's exactly what happens with these megakaryocytes. They die and they shatter into a bunch of pieces. And that's essentially what makes your platelets. So when you have an injury where you have cut yourself and you're bleeding, what happens is the injury will cause your body to release a special hormone. And that hormone is going to cause these fibers to start to anchor on the sides of the injury. And once those fiber threads are in place, they basically create a net. Cannot see it with the naked eye, but it's there. And that net is now in place. So as blood continues to come out of that injury, those fragmented pieces of cell are going to get trapped up in the fibers of the net. And as that happens and more blood continues to come out, more of those platelets get packed into those fiber threads, into that netting. And eventually they get so packed in, they stop the flow of blood. They will eventually harden. That's what you guys know to be a scab, and they will prevent further bleeding. Now, some people get impatient with a scab, they'll eventually pick it off. And what happens when you pick off a scab? It starts to bleed again because you pulled out that plug that was stopping the bleeding, and now blood is going to come out again. So your body has to repeat the process, form a new net, more platelets get packed in, it hardens over again, and you'll continue on until the injury is healed. Now, a couple of things you might have noticed if you get a straight edge cut. So let's say you're shaving, you cut yourself with a razor versus a cut where you fall down on the concrete outside. Concrete cuts are usually going to be jagged edged cuts. And in that case, the fibers that are going to form that net are able to find places to anchor much easier. And so they will get that net in place much faster, platelets get packed in, and the bleeding will stop sooner. A straight edge cut like a razor, much harder for those fibers to find a place to anchor in and get that net started. So you'll notice that those tend to bleed for a lot longer, just because it takes longer to get that net in place. So that is just a little bit about the different types of components that you have in your blood. The last thing that we need to talk about are the blood types and the Rh factor. And there are 4 types of blood. You have type A blood, type B, type AB, and type O. Type O blood is the most common. Type AB is the rarest type of blood that there is. Now, when we talk about the blood types, you have these special features on the surface of your blood cells, which are going to indicate what type of blood you actually have. And so I did a very crude drawing, but I just drew 4 cells and I identified them as being type A blood, type B, type AB, and type O blood. So on the surface of a type A blood cell, you would have a little antigen. It's like a flag indicator, and it would be identified as type A, or that A antigen. Type B blood would have a different little flag indicating that it's type B blood. AB would have both of them, and type O has 0 or none of them. And so that's why we refer to it as type O blood. Now, these surface antigens are going to flow through your body And as other cells bump into these, they will recognize that this is something your body made. That antigen is going to indicate this came from us. Okay. Now, one of the things that we need to know about blood types is if you are going to donate or receive blood, who are you able to donate blood to? Who is able to receive blood from you? And it's all about recognition. So in blood transfusions, when you are taking blood from one person and giving it to another, the exchange you have to make sure that that antigen is going to be recognized by the person you are giving the blood to. So with that marker that you have on the surface of the cell, and you take blood from one person and put it into a second person, they can recognize that antigen, it's going to be a successful transfer. But if you take blood from one person and put it into a second person and they can't recognize the antigen that you're giving them, their body is going to attack it, thinking that it's a foreign substance trying to harm them. And now you're going to have a huge problem. So the antigens on the surface of the blood cell are how we recognize whether or not that blood type is supposed to be in our body. Now, the second piece that I put here are the antibodies, excuse me. Antigens are stuck. They are attached to the surface of the blood cells. Antibodies are not. They float around the blood cells, but they are not physically attached to the blood cells themselves. So here's what happens. These antibodies are trained to attack something that they don't recognize. So you'll notice that type A blood has a B antibody. So if I have a type A person and I take type B blood and I try to put it into my type A person, they're not going to recognize that antigen. And because they can't recognize it, they're going to tell their antibodies there's something foreign here. And that antibody, you'll notice, has 4 little arms. So what it does is it goes and it finds all of the cells that have that B antigen and it grabs onto them and it locks it into one of those arms. It'll go and find another one, locks it into that one, locks it into that one, locks it into that one. So this antibody will grab onto anything that has an antigen that this person doesn't recognize. It's called agglutination. You're taking all of those blood cells and you are gluing them together, which now takes that blood and turns it from being a liquid to basically something the consistency of Jello. So they're not individual little cells all going around on their own anymore. They're now these big, massive globs because they're all getting stuck on this antibody. So the process of agglutination can be fatal because the heart is not designed to pump something the consistency of Jello through your body. And so if they can't reverse it fast enough, the heart basically gets overworked and it will quit functioning. So the type B blood is going to have the A antibody. So if you take a type A blood and put it into a B person, it's not going to recognize that antigen. And if it can't recognize that antigen, it's going to tell those A antibodies, go and find any cell that has an A on it, grab onto it, and keep it from circulating through our body. Your body is reading this as you have a foreign invader. It sees it as something like a virus or a bacteria that is trying to harm you. And so it's going to tell those antibodies, grab onto them so that it can't invade us any further. So with a type A person, if we took type A and put it over into an AB person, we have an A antigen here, we have an A antigen here. They would recognize that A antigen, and that would be successful. But in the case of going from an AB person and trying to put that into an A person, while it would recognize the A antigen, it's not going to recognize that B antigen. And so now it's going to send those B antibodies into alerts, tell them to find anything with a B on it and grab onto it. Now you'll notice here that AB does not have any antibodies. So you could take A, put it into an AB, would recognize. You could take B, and put it into an AB and it would recognize. You could take type O and put it into an AB, because look at the surface of type O. There's no antigen on the surface. So type O is not going to trigger any type of response. There's not anything here for this person not to be able to recognize. So in this particular case, type AB is known as the universal receiver. They can take blood type from anybody. There's a little asterisk on that. But they can take blood from the other 4 blood types and be okay. Type O is the universal donor. Because they don't have any surface antigens, they are not going to trigger any type of immune response if you take type O and put it into any one of the other blood types. Now, the asterisk on that is going to come from the last slide that we have, and that is the Rh factor. The Rh factor is a protein that you have on the surface of your blood cells. And you'll hear people say, I'm A positive or I'm B negative. And the B or the A, the letter part of it indicates their blood type. The positive or negative indicates their Rh factor. If you are identified as being positive, that means you have the protein. So you are Rh positive. If you don't have the protein, you are Rh negative. There is no benefit to having the protein or to lacking the protein. It's kind of like hair color. You know, there's different varieties. You have one, but having black hair over red hair, there's no advantage, disadvantage. So health-wise, you don't have to worry if you're positive or negative, but blood donation-wise, it does become a factor. Now, the reason that it's called the Rh factor is because it actually was first found in a certain species of monkey known as the rhesus monkey, and it's spelled R-H-E-S-U-S, the rhesus monkey. Because that's where the original discovery was made, they just took the first 2 letters from the rhesus monkey name, and it is the Rh factor. So if you have the protein on your blood cells, you're Rh positive. You don't have the protein, you are Rh negative. But it does throw a little bit of a hiccup in blood donations, because a positive person, so if you have the protein, you cannot donate to a negative person. Again, it has to do with the recognition factor. If you have the protein and you try and donate blood to somebody who does not, they are not going to recognize that protein. They're going to tell their immune system that they are being invaded. Their immune system is going to grab onto it and try to destroy it. A negative person can donate to another negative person. A negative person can donate to a positive person, because again, it's kind of like the type O blood. If you don't have the protein, there's nothing there to trigger an immune response. So a negative can donate to another negative. A negative can donate to a positive. A positive cannot donate to a negative person. So there's another little part of blood exchanges that you have to be concerned about. In your lab activity, we are just doing blood types. You are not doing anything with the Rh factors on this one, so just do your exchanges. Don't worry about that. I apologize for the 3 videos. Somehow I managed to hit the stop button in the middle of that second one. It was not my intention to have 3 lecture videos on this particular set, but we did need to get through the blood information. So you have the first video that is going to go over the structures of the cardiovascular system. You have this video, which I apologize, got broken into 2 parts, but it all covers the blood. And then you have a very short video, which is going to be the directions on the blood lab. So I tried to simplify and streamline the lab for you. If it doesn't work, because it's very hard to not be able to show you guys and have to just simply talk you through it. If you have to follow the directions as they are on the lab, that is fine. But if you are able to pick up the change that I made, it's going to save you a lot of time and a lot of cleaning. So do what you need to do, send me questions, but just so you have that information as well. Take care, and I will see you next week.